Hi everyone, I'm Jaya Vishwanathan and you've found the No Brainer podcast where I discuss all things neuroscience in delightful conversations with various experts. I hope you enjoy today's episode.
Hello, everyone. Welcome back to another amazing episode of the No Brainer podcast. I am very excited today to introduce you all to Dr. Paul R. Territo, one of my mentors, and I'm very excited that he's going to be able to share some of his wisdom and scientific expertise with all of us today. So Dr. Paul Territo is a professor of medicine at Indiana University School of Medicine. Dr. Territo received his bachelor's in 91 and master's in 93 from California Polytechnic University in Pomona, California, and then his PhD in 1996 from the University of Nevada, Las Vegas. During his postdoctoral fellowship at the Laboratory of Cardiac Energetics at the NIH's National Heart, Lung, and Blood Institute, or NHLBI, he received medical retraining in the areas of non-invasive imaging technology and biophysics, which included positron emission tomography, PET, computed tomography, CT, and magnetic resonance imaging, MRI. From that time forward, his research has focused on the development and application of imaging technologies to study disease progression and evaluate the role of novel therapeutics on our biological processes. Leveraging these experiences from 2001 to 2008, Dr. Territo was a part of Eli Lilly's drug discovery team and developed novel imaging-based biomarkers for therapeutic development in neuroscience, oncology, and cardiovascular disease. Shortly thereafter, Dr. Territo joined the faculty of Indiana University School of Medicine in the Department of Radiology and Imaging Sciences, where he served as the director of the preclinical imaging core. Over this interval, Dr. Territo continued to develop novel imaging-based biomarkers for both preclinical, and he'll be able to explain what that means in a second, as well as clinical assessment. In 2016, Dr. Territo began his independent laboratory, focusing his research on developing state-of-the-art biomarkers for monitoring disease progression and then leveraging these for assessing therapeutic response for Alzheimer's disease and related dementia. So with this storied career and biography, you can see why I am so excited to have Dr. Territo on. And I have so many questions for you, so I'll try to be a little bit more focused myself because I know I get very enthusiastic every time I chat with you. So I'd like to start off with this question. So you are a leader in multiple fields like biomarker discovery, bioenergetics, which I'm very interested in personally, drug discovery and development, non-invasive imaging techniques, which is really important in human research to understand both brain and cardiovascular function, among others. So can you tell us a little bit about your unconventional journey and what you enjoy about your work?
Yeah, sure. First off, Jaya, thank you so much for having me on your show, and also it's a real pleasure to get a chance to speak with you today. So my journey into medical research was a fairly non-conventional one. I did a bachelor's, master's, and PhD largely in basic science, and this was a good experience and lots of fantastic opportunities to learn new areas of research. The areas that I tended to focus on during my master's and my PhD was in comparative physiology. So comparative physiology is where you look across either multiple species or multiple biological organizations. So if you think about looking at whole organ or whole organismal level readouts and then paring those down to more and more reductionistic measures. So think about an organism, the heart, the cells that make up that, the organelles that make up that cell. All of that level is also considered comparative. And so that was a great training ground for me. Lots of interesting research. At that time, I was working largely in the cardiovascular and respiratory system. And it was a great launching pad to go to the next phase of my career. At the end of my PhD, I applied for multiple postdoctoral positions and was fortunate during one of those to actually land what's called an IRTA fellowship at the National Heart, Lung, and Blood Institute. This is a intramural research training associate fellowship. The fellowship was designed to actually take basic scientists and to retrain them to perform translational medical research, because at that time there was a dearth of researchers in this space, and so they were looking to add new scientists in this space and help fill that gap. And so I did a fellowship in the laboratory of cardiac energetics. This was Dr. Bob Balaban's lab. And I spent 5 years there and really enjoyed my time, loved doing the research. We were doing a lot of research around heart function, heart failure, and then what are the factors that were driving those processes. And in particular, my research was largely focusing on the energy control in the heart. And this is where the bioenergetics piece comes in. So bioenergetics is really looking at how you take substrates and follow their path through the biochemistry, ultimately to produce energy so that the heart can perform its function. So if you think about, your daily life, you get up and you walk across the room and your heart just beats away and it does its thing, and you don't really think about how that energy is being supplied to that muscle. And so I was looking at how is the heart energy controlled, and how things like calcium, as an example, were a primary driver of those biological processes. And at that time, our understanding of how energy was controlled in the heart was pretty thin. There was a good understanding that the biochemistry around energy utilization in the heart was controlled through the TCA cycle, the tricarbolic acid cycle, or the Krebs cycle. So, you take in substrates in the form of food, they're broken down by enzymes in your digestive system and ultimately absorbed and then taken up into cells. But what wasn't known is how do you actually turn up the volume on that energy flow as the heart needs more work or performs more work. And so one of the things that we were studying was how calcium can actually drive that process. And it's this exquisitely coordinated process that, as the heart begins to increase its contractility, the amount of force that it's generating because it's trying to move more blood, obviously it needs more calcium to do so. And in the process, it turns out that calcium signals to the organelle on the cell, the mitochondria that produces all that energy, and it turns up the processes in a very non-linear fashion. And it turns out it's very important for understanding how the heart maintains its function, not under normal conditions or even workload conditions like when you're running a marathon, but it also turns out that it's very important in terms of disease states, because as your body undergoes a process of disease, of course, the relative amount of energy that's used to not only perform function goes up, but also the amount of reserve that you have that you can use for these other processes, like running across the room, goes down. And so that functional reserve is depleted, and as a result, you actually need to have a very well-tuned system that can handle those kinds of conditions. So after my fellowship at the NIH, I applied for multiple positions in the pharmaceutical and biotech arenas, and was fortunate to work in a small biotech company in Vancouver, British Columbia. This work was interesting in that I was able to now take some of the new training that I had received and apply them in a more real-world setting. And this is work that was actually looking at inflammation and trying to understand how we can develop pharmaceuticals that are not the traditional drugs that we think of on the market, like aspirin or even Tylenol as an example. These kinds of drugs were known and have been around for a very long time, but we were looking for that next generation of anti-inflammatory drugs. I was there for about a year, and one of the things that occurred over that time was I had a lot of learnings. Some of it were about the importance of doing good scientific rigorous research. Some of it had to do with trying to navigate the wilds of living outside of the country when you were born in the United States. And some of it had to do with, personal things like having a family and starting a new life, and having children, of course, also means that you need to be closer to home and closer to family. And so that facilitated us needing to move back to the United States, where I started applying for positions. And during that time, I was fortunate to join Eli Lilly's drug development arm, which is called Lilly Research Labs, and took a position there. And my initial position was in the veterinary medicine research group that actually performs and develops drugs for companion animals, cats and dogs. And so I worked in that group for just about 2 years. And since that time, it's been really interesting because a lot of the drugs that I worked on while I was there have now come to market. And several of these are things that you can purchase on the shelf right now. Some of them are things like drugs for anxiety or depression, separation disorders that you have in dogs and cats when you leave home and you leave your pet at home by themselves. Some of the drugs that I worked on were for flea medications, as an example. So, so it was a great learning experience, and had a really great time working there. But at that time, the human division of Lilly offered an opportunity to move into an area that they were just starting to build, and this is utilizing medical imaging as and biomarkers as a readout of drug efficacy. And so this is where you kind of come back to your home roots. And for me, having been trained in this area, and now being able to apply this to a more complex system where you're looking at the biology and the drugs and how they're interacting, this was really appealing to me. And so I ended up joining what's called Lilly Research Labs, which is the group that actually develops pharmaceuticals for human. And I was in a group that developed these imaging-based biomarkers for their team. I think I was there for just under 10 years. And it was a tremendous, exciting time because a lot of the things that we're going to talk about maybe later today, about some of our research and some of the things that we know about drugs for Alzheimer's disease, were just starting back then. And my lab was working on many of the biological readouts as measured by medical imaging, and how we could actually help inform the clinic with those. Yeah, so it's been a fairly non-conventional journey, passing from basic science through medical retraining and then into the pharmaceutical arena. Yeah. And I was, like I said, I was in pharma for a good while. And unfortunately, somewhere around 2008, late 2008, there was a downturn in our economy. And that had a pretty negative impact on pharmaceutical research at that time. And many companies, including Lilly, for the first time had to lay off employees. And that was, a difficult decision by some of the leaders in those companies. And I know it was at Lilly because it was the first time that Lilly had ever laid off an employee in their almost 100-year history. But I was amongst a group of 800 people or so that were laid off. And so what Lilly did was they actually arranged so that the groups like ours that were working on very important aspects of their drug development pipeline were able to find positions in groups like contract research organizations. Contract research organizations are essentially the fee-for-service arm, if you will, of the pharmaceutical industry. So in some cases, it's doing a little bit of discovery research and helping you understand things about your molecules. Sometimes it's performing, in-life studies in animals. And in some cases, it's actually performing some clinical trials, because these companies span the entire spectrum from beginning to the end of how a drug makes it through its lifespan. I was at a contract research organization called Covance. It's now been purchased by another company, but I was there for about a year and ran the nuclear medicine division for that group. And interestingly, during that time, our group was actually still providing the same research back to Lilly. They had contracted, because we were there, because my group was there, they contracted the work directly to us, which was great for both the company and as well as myself, because we're still working with colleagues, just in a different format.
Right.
About a year or so into that, I was approached by the School of Medicine Department of Radiology, and in particular because we had done some work with them, and so they asked me if I'd be interested in coming to help run their preclinical imaging group. Now, preclinical imaging means essentially the stage of drug development process prior to the clinic, and then non-clinical, which is the phase right before the clinical approval of a drug, and then clinical research. And so clinical research ultimately leads to the approval of a molecule. So the preclinical phase, sometimes if you talk to a clinician, they'll intermix preclinical and non-clinical, but they essentially are slightly overlapping processes. Is the phase of research where basically most medical schools are performing their studies. They're doing basic science questions applied to medicine, so asking scientific questions in a medical context, but they're doing it primarily in animals, and they're doing it to help further our overall understanding of either the disease process or the therapeutic that they're working on and the mechanism by which it works. That's very similar to the work that I was doing when I was working at Lilly and also at the Contract Research Organization. And so it seemed like a natural fit. It was very exciting to think that I could join a group and make a difference. And so I joined the group at Radiology. I think it was in March of 2010. So I've been at IU ever since. So unlike most people who have a very linear path in their career, where they go from an academic training and then to a postdoc and then to the next phase, which might be an assistant professorship, I kind of took this long route, which gives me kind of a unique view of the world, because I not only have an appreciation for the basic sciences and how those contribute to our understanding of medicine, but also how those are used in a drug development paradigm and in a clinical paradigm as well. So our lab is kind of unusual in that we like to think about the work that we're doing as translational in nature and how it impacts patients' lives. And when I was running the core for many years, I was doing that for other investigators. So I was helping to support their work. So it's and somewhere along the ways, you kind of start to get your own ideas about how you would like to conduct your research. And you have your own ideas about how to test the questions that you think are most important. So that's where my lab started.
I just wanted to say I loved how your trajectory also just tangentially highlighted like how animal research can help improve the quality of life of not just humans, but also animals. And I think that's an often overlooked aspect of, when we're in this space of trying to just increase our general understanding of how the body works and how the brain works. And it's just we're adding to our collective knowledge and helping these therapy development processes in many ways. So thank you. So there are a lot of different things that I kind of want to dig into, which is very fascinating, again, about your trajectory. So I'm going to skip ahead to maybe because I would like to focus a little bit on what you're currently working on Alzheimer's, maybe a little bit later in the conversation.
Sure.
So maybe we can start off a little bit for people who are maybe unfamiliar with pharmaceutical interventions and how they might be working. So can you talk a little bit about what are the different kinds of things? So you mentioned already like the non-inflammatory NSAIDs, anti-inflammatory drugs, but then there's different kinds of drugs that people hear about. Some are injectables with proteins, and how does that work with something complicated like the blood-brain barrier, which we talked about actually a little bit in another episode as well. But just kind of an overview of like what are the different kinds of pharmaceutical interventions that people are kind of working on these days?
So pharmaceutical interventions can be sort of primarily broken down into what we call large and small molecules. So those two categories are namely the small molecules, or sometimes what we think of as synthetic or semi-synthetic molecules that are produced in the laboratory by a chemist. They're organic molecules. So these are molecules that are starting with simple building blocks, and they're built up into a more complex molecule that allows that molecule to interact with the biology in a way that will give a biological effect, a desirable biological effect. When they don't give the desired biological effect, sometimes we call those toxic molecules or toxicants. But in the case of a pharmaceutical, it's a molecule that will drive it in a intentional direction that improves the function. So you can think of it as a pendulum. The whole process is a pendulum, where the disease process takes you away from the bottom of the arc of the pendulum, and where you move more towards a disease state. And the pharmaceutical will move you back towards that central point. If you give too much of the drug, what will happen is you'll swing on the opposite side, and you end up getting sort of the toxicity of the drugs that we oftentimes hear about or discussed, when you hear these molecules listed on the television or a radio commercial. There's a whole list of side effects. So those can happen with small molecules in particular because they're small. They only bind to a small portion of the targeted protein that they're interacting with. And as a result, that little shape that it's binding in, this three-dimensional pocket, they call it, is not just unique to that particular protein, but they're found all over the body. And it turns out that that's both advantageous, but it also can be disadvantageous because sometimes you get what we call off-target effects or untoward effects of the drug interacting with these unintentional locations that you didn't want the drug to work on. So that class of molecule it has some advantages. It's easy to manufacture. It's relatively low cost. You can make very potent molecules that bind to very specific parts of proteins. The other class of molecule is called a large molecule, and these are, again, either synthetic or semi-synthetic molecules. The way that they're synthesized, though, is way different. You're not using simple building blocks of organic molecules to build them. You're basically making an organism, like a bacterium, express a gene of interest. And the way you do that is might be beyond the scope of our conversation today, but essentially you're using some genetic engineering. You're inserting a piece of a DNA that you want the bacteria to produce. During its normal operation, the bacteria will then read that DNA and start producing that protein of interest, and it produces in large quantities. If you think about it, it's kind of like a bioreactor. It produces lots and lots of the material. In most cases, these are proteins or antibodies. And then because the bacteria is much larger than the antibody, they can filter out the bacteria. And then, of course, you can get a nice purified antibody that's very specific. These kinds of molecules are really interesting because they're more closely aligned to the natural ligand and receptor interactions that you would expect to find in the animal, or in the person. And so if you think about it, proteins are really three-dimensional objects. They're small peptides that are folded up on themselves, and they have a very distinct shape. And what's interesting is that these large molecules are more specific about where they bind on those shapes. So instead of just binding to a little tiny part of that protein, which it might be expressed all over the body, these large molecules, antibodies or peptides, can bind to a much larger fraction. And as a result of that, they're much more specific about where they bind and when they bind. And that gives them the advantage of selectivity, and it also gives them the advantage of specificity. So as you might imagine, we'll reduce that side effects that we were talking about with the small molecules. So there are some things about these molecules, though, that make it more challenging, and that is that they're hard to manufacture. They have higher costs. So those are the basic kinds of pharmaceutical.
Thank you for... Yeah. No, thank you for making that so intuitively easy to understand, because I think a lot of the time when you see all the things that are happening in this space, it can very easily start to overwhelm you. So thank you. That pendulum example was fantastic to kind of help people understand normal, disease, and then off target. Thank you for that example.
One of the things that is really interesting about these classes of molecules is that they, because of their size and complexity, they have different penetration into the brain. So for instance, small molecules that are below around 400 grams per mole, or it's a weight essentially of the molecule, they tend to be reasonably oily like membranes are, because membranes are made up of phospholipids. Then they tend to penetrate into the brain pretty easily. And so small molecules can get usually into the brain without much trouble, delivered by the bloodstream. Large molecules, on the other hand, a little more challenging because they're large. First off, they're on the order of 10,000 in terms of molecular weight. So that's grams per mole. And so as a result, they don't necessarily want to diffuse through the membrane very easily. And so only maybe about 0.1% of most molecules that are large molecules like antibodies get into the brain, which makes treatment with them quite challenging. So if you think about, Alzheimer's disease, like we were talking about, most of the currently approved molecules that are these in this class have this limitation, and they struggle with getting enough drug molecules into the brain to get to the target of interest to give the biological effect that they're after.
Right, and I imagine like figuring out the techniques to target them also adds to the manufacturing costs, which makes it, and then there's a whole thing about how insurance covers for it, which is definitely outside of the scope of today's conversation, but that makes a lot of sense. Yeah. So kind of an interesting segue from therapy development is this idea of biomarkers, and especially therapeutic biomarkers, diagnostic biomarkers. And I know there's all these different kinds of biomarkers that you can use for different stages of the disease. But then I kind of want to talk more about what your research on developing these novel biomarkers in translational, given that you're really in this frontier of discovering more about how our bodies and brain work and then how to quantify these. So specifically, if I might ask you to kind of focus a little bit on also just the possibilities and challenges of working in biomarker discovery. I think that will be very insightful for people tuning in.
Yeah. Sure. So biomarkers can be stratified across a number of different areas. It's a term that's used pretty loosely, although in the world of regulated research like the FDA, that has a very distinct meaning. A biomarker is a well-controlled, well-measured, understood single measurement of a particular readout in the biology of an organism. But in the general sense, I think biomarkers have really, taken off over probably the past 30 years of research. The ability to measure different aspects of the disease process has really grown exponentially, and this is in part due to the technical advances that have occurred in these areas. There's also been a fair amount of investment by both the federal government as well as regulatory agencies, because they understand the value of these particular markers and their ability to not only follow disease, so as a diagnostic, if you will, but also as a readout of the therapy and how it's changing that biology, that disease biology that we were talking about before. Mm-hmm. For instance, one of the areas that's really exciting and hot right now is fluid biomarkers. These are used a lot in Alzheimer's research, for instance. You can measure these in the blood or the cerebral spinal fluid. This is the fluid that's produced by the brain and is filtered through the spinal canal and ultimately gets back out into the bloodstream. Mm-hmm. But if you think about those fluid compartments, they actually contain small molecules. Those molecules are an indication of a sample of what's going on in that compartment that we're interested in. And so, these markers allow us to actually follow the disease process. We call them analytes. And over the past years or so, the growth in this area has just been extraordinary because we can measure analytes using new technology like mass spectrometry. This is an approach where you inject, in this case, blood into an instrument. The instrument will then separate the molecule from that fluid compartment that it's in, and it'll look at the mass of it and the charge of the molecule and be able to distinctly tell you what that amount of that mass and that charge was for that particular molecule. So that technology, which primarily was used 20 years ago only for small molecules, has now been expanded into large molecules like proteins. And so this has given us the opportunity to measure analytes all the way down into the sub-nanomolar concentration range. So we're talking about molecules that are on the order of nano to picograms per mil of analyte per fluid volume, which is really amazing. If you think about that, that's an amount that you couldn't visualize, obviously, but it's there, and they can actually see this. Yeah. And so these are great because they allow you to get a snapshot in time of what's happening in these compartments, so in the blood or in the cerebral spinal fluid. The only downside is that they're spatially and temporally distinct from the compartment you're really interested in this case, the brain, for instance. And so that makes it a little challenging because these compartments, although they're in dynamic exchange with each other, the blood and the brain and the CSF in the brain, there is kind of a lag in time. And so, the time in which you actually make the measurement may not be the actual time in which the event has occurred. And so there's a little bit of, I guess, an understanding that you need to have about that temporal shift, if you will, in the readout of your measurement. But that's really an exciting area. It is something that is really taking off right now. In fact, there's a group out of Sweden that I think probably produces a paper a week on brand new analytes that they found in, this is a Zetterberg lab, and they produced just an enormous number of publications on brand new analytes that they find in patients who have Alzheimer's disease relative to those who don't. And so, and they're exploring these as potential early markers of the disease process. The other kinds of markers, I think, that fall into this category are things like medical imaging, which is an area that my lab spends a lot of time thinking about. And this includes MRI that you talked about earlier, and PET imaging. And these have advanced our understanding, at least in terms of structural and functional changes that occur within the brain. And those are non-invasive. MRI largely gives you anatomical changes, although there are some specialized techniques that can measure things like diffusion or perfusion quantitatively. But generally, as a whole, MR is a anatomical measure. And in the early days of Alzheimer's disease, how we determined when someone had it was not only looking at the brain and its shrinkage, but also looking at the centers of the brain that are responsible for short-term memory, like the hippocampus, and its shrinkage. You might imagine that has to occur at a molecular level way earlier than the actual structural change that you're seeing in the MRI. And this is where technologies like PET actually have an advantage, because PET imaging is taking some of those small molecules that we talked about earlier, and these are basically just pharmaceuticals that we've labeled with a small amount of positron-emitting radioactivity, very short-lived material, but very high energy. And so we can actually detect where that goes in the body because it binds to a protein, a target of interest, and allows you to quantify the exact amount of that at that location. So in the case of Alzheimer's disease in the early days, we used it to measure things like glucose metabolism in the brain, and then ultimately things like amyloid deposits in the brain. More recently, they've been doing things like tau deposits in the brain. But it's a very quantitative, very highly sensitive method, and it's non-invasive, which is really the key thing, because we can see where it is and we can see how much is there. They do have some downsides, though, just like everything. Everything's got an up and a downside. The downside here is that they're a little expensive. I think, as anyone who's ever had an MRI or a PET scan knows, they can be in the thousands and thousands of dollars, tens of thousands of dollars in some cases. And oftentimes they require some secondary endpoint measure to confirm the biology.
Right.
So you might come in, let's say you had a blood test or a cognitive test, then these imaging methods would be the second line of confirmation that we say, yep, we can see you've got deposits of amyloid in the brain, or we can see that metabolism is depressed in the brain, and so you must be on the trajectory towards Alzheimer's. And there's a whole nother class that is just recently coming out, and I'm sure you're probably aware of this, with everyone having smartwatches these days. Digital biomarkers are another area that's really taking off. And I know when I was just leaving pharma back in right around 2010 or so, companies were really investing heavily in some of these technologies. They were looking at things like wearable devices that will allow you to explore things like motion or gait of a patient. And because they can measure things continuously over long periods of time, they allow you to collect data that is pretty low cost and pretty continuous, which has an advantage because oftentimes scan is a pinpoint measurement in time, much as the fluid biomarkers are that we talked about. But digital biomarkers give you a more continuous readout. The only downside of those is that they are relying on other technologies to confirm this integrative response, because you're really looking at things like motion, right? A patient walking and their lack of cadence, so dyskinesia, if you will, And dyskinetic motion is really a lack of integration at the brain level to communicate to the muscles to move appropriately. So that's a whole lot of steps that have to go between where you're thinking about the process and where you're translating it into motion. So ultimately, these biomarkers haven't found their way as the primary readouts of disease or therapeutic response, and it's removed from how a drug works, although they do lend some clues as to what's going on.
Right.
Hey everyone, hope you enjoyed this episode of the No Brainer podcast and see you at the next one.